So our next biotechnology topic is recombinant DNA. Recombinant DNA is DNA that has genes from multiple species. So we can put a fish gene into a plant, or we can put a fungi gene into a bacteria. This allows us to create what we call transgenic species. So a transgenic species is a species that has DNA from a completely different species. This comes from genetically modifying organisms, but specifically now we're putting different DNA into them. So how does this actually happen? First, you identify a gene of interest from a particular organism, and it gets spliced out of the organism that normally carries it. And that individual gene is then spliced into something called a plasmid. And a plasmid is a very small circular piece of DNA. Plasmids are particularly good at moving around and invading different cells. So you can expose bacteria, for example, to a plasmid, and the plasmid will carry that little circle of DNA, including the desired gene, into your bacteria or other target organism. Why would you do this? There's a couple different reasons. You can use this process to identify gene products. So let's say there's a gene and you're interested in exactly what it does in humans or in dogs or in cats. You can extract it from a cell. You can create a transgenic bacteria, and that bacteria will now produce a particular protein. This allows you to infer that the source of that particular protein is that particular gene. The other common way this is used is to produce large quantities of specific proteins. So this is the process that's used to make penicillin, for example. The protein in penicillin is actually produced by various kinds of fungi. One of the best ones is the green mold that grows on the outside of cantaloupe. But it's hard to harvest just from cantaloupe, and especially to get sort of medical grade level penicillin. So instead, they've figured out the specific gene from the fungus growing on the cantaloupe. They have spliced that out and inserted it into bacteria, and you can tightly control how the bacteria grow. The bacteria contain this gene, they go through transcription, translation, they create the protein, and you can just siphon off all of the penicillin that's being produced. It's not unlike having a dairy where you've got cows making milk, except now we have individual bacteria producing specific proteins for us. So you're going to watch two videos now that show this process of using a plasmid to move DNA around, and how that's used in a medical application to actually produce large amounts of particular proteins.